Namaste, dear students. All are welcome to our biology class. There is a lot of living things on this earth: birds, animals, plants, humans, reptiles, and so on. Then, all organisms' body is made up of cells, isn't it? We already studied about the structure of animal cell, structure of plant cell, and what are the differences between plant cell and animal cell. Okay, then, but we cannot see these cells with our eyes. We need a device for this, for magnifying the object. What is the name of that device? Yes, it is microscope. We need to know more about microscope in order to observe a cell through a microscope. So today we will get to know the parts of microscope and their uses.
Compound microscope. Objective: to learn about the construction and working of a compound microscope. A compound microscope has multiple lenses which collect light from the object and then focus the light into the eye. These microscopes have the ability to magnify very minute objects to a substantial size, clearly showing the smallest details. There are three major parts to the compound microscope: a body tube, an objective lens, and an eyepiece. The body tube is comprised of two smaller telescoping tubes with lenses coaxially mounted at either end. The wider tube holds the objective lens, while the narrow tube holds the eyepiece. The knobs for both coarse and fine adjustment allow the tubes to slide one into another, varying the distance between the two lenses. The lenses are convex with different focal lengths. The lens closer to the object is the objective and has a short focal length, FO, usually below 1 centimeter. The lens nearer the eye is the eyepiece. Although the eyepiece also has a short focal length, FE, it is still greater than the focal length of the objective. A revolving nosepiece, known as a turret, is attached to the lower end of the objective. The turret can be rotated to change the power magnification as necessary. Below the objective sits a flat platform known as the stage. On the surface of the stage, two stage clips hold the slide in place. A small aperture is present in the stage, allowing light into the body. The body tube is arranged on an arm which is connected to a strong base. This arm allows the body of the microscope to tilt. A small mirror at the base of the microscope reflects the light through the stage aperture. Above the mirror, an iris, also known as a diaphragm, is present. Iris helps in controlling the amount of light passing into the stage aperture. Working of the compound microscope. The object O is placed just outside FO, the focus of the objective lens. A real, inverted and magnified image, I1, is formed. The magnified image, I1, acts as an object for the eye lens. The eyepiece is adjusted such that the image I1 lies within the focus of the eyepiece, FE. The eyepiece functions as a magnifying glass and forms a virtual, erect, and magnified image, I2, of the object. Magnification of the compound microscope. The magnifying power of a lens is expressed in diameters. A lens magnifying an object 5 times has a magnification of 5 diameters and is written as 5x. The total magnification of a compound microscope is the product of the magnifying power of the objective and the magnifying power of the eyepiece. For example, if the objective has a magnification of 40x and that of the eyepiece is 10x, then the magnifying power of the microscope is 400x. Summary. The compound microscope has a system of convex lenses allowing the magnification of minute objects to that of a very large image.
Okay, children. Now you understand the parts of microscope and functions of its each part. Here is the picture given in our textbook. All of you label the parts of microscope. In this picture, there is space given for writing the name. All of you write that. Okay. Okay, children, our next activity is preparation of observation material. Actually, the preparation of observation material requires much accuracy and patience. Okay, the steps for the preparation of observation material is given in our textbook. All of you read it. And before that, let us watch a video for better understanding. Okay.
Characteristics of dicot and monocot stem and root. Stems and roots of plants are made up of different types of tissues. These tissues form different layers in the composition of stems and roots. To study the structural details of the stem or root of a monocot or dicot plant, it is essential to get familiarized with the sectioning and staining techniques used with plant materials. It is also necessary to take the sections with uniform thickness so that the light passes through them equally and the different tissues found in the material are clearly visible under the microscope. To examine the tissues clearly, it is desirable to stain the section with suitable stains, as different stains color the tissues differently. Materials required: fresh material of sunflower stem, maize stem, sunflower root, maize root, new blade, compound microscope, slides, cover slips, petri dish containing water, saffron in solution, glycerine, brush, needle, filter paper, dropper. Procedure: take a blade and a sunflower stem to be sectioned. Dip the top of the material and the blade in water. Hold the material to be sectioned horizontally between the thumb and the first finger of your left hand. Cut the sections of the material quickly using the blade. Transfer the sections in petri dish containing water. Using a brush, select a thin, uniform and complete section and place it on the glass slide. Using a dropper, take some saffron in solution. Pour 1 drop of saffron in solution over the section and allow it to stand for 2 minutes. Remove excess stain using a filter paper. Pour a drop of glycerine over the section. Place a cover slip gently on the section with the aid of a needle. Remove the excess glycerine using a filter paper. Repeat the same procedure for the sunflower root, maize stem and maize root and prepare the slides. Observe each section under the microscope. Observations: dicot stem.
Okay, children, we can observe the plant cells through the microscope. That is the observation. Then look at this picture here. The magnification power, that is 10x and 45x. What is the difference when we placed the material or the specimen under 10x power, and what is the difference when it is placed in the 45x power? Okay, in your textbook we already said about the stages of preparation of observation material. All of you write the stages in your notebook and study well. Okay, thank you.